Welcome, everyone. I'm Dr. Carmen Pugliafito, and I have here today Dr. Jayakrishna Ambati, who's the Guerry Professor at the University of Virginia Medical School. Jay, welcome to Retina Synthesis.
Carmen, thank you very much for the kind invitation, and I look forward to speaking with you. And it's always a great pleasure to be with you.
So, you've done a lot of innovative work in the field of retinal biology and clinically relevant models. You're working now on treatment of geographic atrophy using the inflammasome target. Can you just discuss the basic biology of your treatment approach and why it might be better than complement inhibition for geographic atrophy?
Yeah, sure, absolutely. I'd be delighted to do that. If you would permit me to give a little bit of background on it, because this has been really a team effort that really stems back now 20 years. I've been blessed to have an amazing team as part of my group, first at the University of Kentucky, where I started my faculty career in 2001, and then continuing here for the last 10 years at UVA. And so my group for the longest time, since the inception of our group back in 2001, has been studying innate immunity. First in neovascular AMD, and then we switched tracks to studying dry AMD because with the advent of anti-angiogenic therapies, obviously dry AMD and GA became the greatest unmet needs in certainly retina, if not in ophthalmology, and one of the biggest unmet needs in all of medicine. So, in our studies on innate immunity, a really talented group of folks, including Ben Fowler, Brad Gelfand, Mark Kleinman, and Valeria Torallo, among many others that I've been fortunate to have with me as part of my group, we, as a group, made this very interesting discovery that a group of proteins called inflammasomes, which basically are if you can think of it, they're sort of like the smoke detectors of your cell. They detect some sort of danger going on in the environment, inside the cell or outside the cell. And that can be in the context of infection from pathogens. It can also be in the context of abnormal proteins that congregate, like amyloid beta in Alzheimer's, or TDP-43 in ALS, or in the context of geographic atrophy, which is really a multifactorial disease. Inflammasomes are detecting a whole array of abnormal signals. That can be complement activation, it can be iron overload, it can be something we've spent a lot of time studying, which is ALU retrotransposons accumulating. It can be lipid dysmetabolism. So there are a lot of different stressors that drive geographic atrophy, oxidative stress, mitochondrial damage. All these things um, are ultimately culminating, it seems, work from our lab as well as many other groups around the world have shown that this inflammasome pathway is sort of a final integrating pathway, a final checkpoint. And so we thought, and it seems to be the case now, that blocking inflammasomes could be a final common stopping point, regardless of what the originating stressor or danger signal is. So that's what we've been focusing on, and now we've been able to test in a clinical trial that's ongoing.
So, what's the inhibition strategy?
Yeah, so we have a small molecule called Kamuvudine K8, and I have to plug in the creator of these molecules, who's also my better half, Kameshwari Ambati, and it's named after her. She's an organic chemist that I was very fortunate to meet and now marry. And she created these molecules along with our group. And so there's a whole suite of these molecules that we have for many different diseases. K8 is the one that we have now in the clinic for geographic atrophy. It's a small molecule that actually targets multiple inflammasomes. So inflammasome is not a single molecule. There's more than a dozen of these inflammasome molecules, and there's a certain sort of redundancy and multiplicity in what they detect. And so it's important to block more than just one inflammasome, and K8 does that. And because K8 has a short half-life, being a small molecule, we have a sustained release device implant that's injected into the vitreous humor and releases drug for about 3 months. And so the design of our ongoing phase 2 trial is that the implant is injected every 3 months, and it releases the drug for that duration of time.
So, what kind of implant is it? What is it made of? What is it made of?
Yeah, it's a bioerodible implant, one that the FDA is comfortable with. It uses technology that's been in people for the last 40, 50 years. We haven't publicly disclosed what it is yet, but basically it's a bioerodible implant that disappears shortly after the drug cargo has been dissolved.
So, this approach basically t targets a final common pathway in the pathogenesis of geographic atrophy. Did I get that right?
Yeah, absolutely. And you mentioned the complement strategy before. As we in the retina field know, there are now two complement inhibitors that have been approved. They have modest efficacy, I think it's safe to say, and they have some substantial adverse effect profile, as the clinical trials, the pivotal trials have shown. It's also important to remember there have been at least 12 other complement inhibitor trials that have failed, that have not met their primary endpoint. And so I think it's safe to say that complement inhibition alone as a monotherapeutic strategy is likely to be suboptimal, and we're seeing that. And we clearly need to do something more. And that's probably not surprising given that we know that GA, like Alzheimer's or Parkinson's or ALS and other diseases, are really chronic multifactorial diseases. I like to think of it as a sort of systems failure. I mean, GA generally doesn't happen when you're 20 or 30 years old, right? It happens when you're 60, 70, 80, 90. And there's an ongoing systems failure if you want to think about it from an engineering perspective. And ultimately, the body's efforts to maintain and repair itself are lost. And there's a capitulation event, if you want to sort of invoke sort of financial stock market metaphors. And it's that capitulation event that we're trying to stop, the death of the RPE and choriocapillaris and photoreceptors and so on. And this is not just being driven by complement alone. And we know that, not only from the clinical trials, but even from basic biology. So it is important to target a multiplicity of factors that are driving this disease. And we think inflammasome inhibition allows us to do that because it is really at the bottom of that trough where all of these signals are coming in, complement and many others.
So, tell us a little bit about the clinical trial results that you reported at the Angiogenesis 2026 meeting.
Yeah, so this is a 6-month multi-center trial based in the United States, and we're very grateful to all the trial sites and the PIs and the clinical trial coordinators, and of course, the subjects that have kindly agreed to participate in this trial. And so I presented data from the interim 3-month endpoint for our first two cohorts. This is a dose escalating study. So we have 10 patients in each of three different dosing cohorts. And what we found in the first two dosing cohorts of 20 patients at 3 months is really rather remarkable. So, as a sort of benchmark, while it's not a head-to-head comparison, but if you just look at the results from the two approved GA drugs, the complement inhibitors, at 3 months, they reported anywhere from 0% to 16% reduction in the size of the lesion expansion. So the area of the lesion size growing was reduced anywhere from approximately 0% to about 16% over 3 months. And what we found in our cohorts is anywhere from 40 to 50%, depending on the dosing cohorts and depending on how you look at the metric of growth. So we're seeing much greater efficacy. The other thing we're seeing is a much cleaner safety profile. So far in these 20 patients, we have not seen the more relevant safety signals like macular neovascularization, vasculitis, endophthalmitis, detachment. We've been fortunate not to encounter any of these complications so far in those 20 patients up to 3 months, actually now beyond. And the other thing we're seeing is not only is there a much greater efficacy when you compare across trials. But the onset of efficacy is much more rapid as well. So if you look at a sort of a clinically relevant metric of how quickly the drug starts working in terms of reducing the front, the GA lesion growth by at least 50 microns. We see that that took about 11 to 14 months for the two approved GA drugs, but in less than 3 months, the K8 implant was able to stop the growth of the front of the GA lesion by more than 50 microns. So we're seeing it works rather rapidly, really right out of the gate. So that's also very encouraging.
Yeah, that's impressive. So in this study, you used the fellow eye as the control. Can
That's
you
correct.
comment about that strategy?
Yeah, absolutely. It's not a novel strategy. It's actually something that several other luminaries in the retina field have recommended for a long time, starting with Janet Sunness and Phil Rosenfeld and others. And at the angiogenesis meeting, as you know, there were a couple of other presentations by Linus Shen and others that have shown how robust using the fellow eye is. So if we sort of step back, the phase 3 trials that the FDA has permitted have looked at one group of patients treated with the active drug and a separate group of patients observed, so-called sham injection, just touching the eye with a needle hub without an actual injection. Now, there are a number of reasons why using the fellow eye is actually a much more robust control. The first is if you look at the parameters, the factors that influence the growth of the lesion. For example, all these internal patient characteristics like genetics or diet or smoking or various other factors, they're obviously identical, right? Medication use in large part, they're obviously identical between the two eyes. But even more important in terms of balance is that if you look at the rate of lesion growth in fellow eyes of bilateral patients, the correlation coefficient is very high. It's like 0.88, zero point 0.9. And that's the case if you look at natural history studies like the ARED study or Blue Mountain, or if you even look at the two phase 3 trials from the two approved GA products, they've reported that in fellow eyes, the difference in lesion growth over 12 months was only 4 to 5%. So the bottom line is there's a very high correlation between the lesion growth in one eye versus the other eye. So that's a great way to compare. The other reason in addition to that, is that the correlation between unrelated patients is close to zero. It's like 0.001. So if you look at two separate patients, as you might imagine, there's virtually no correlation between the lesion growth. That's why you have to use hundreds or thousands of patients to compare. In contrast, if you use fellow eyes with 100 patients, with 50 patients, depending on how powerful your effective drug is, you could make comparisons that are very statistically powerful. So that's why we use this approach.
So visual acuity, that's
Mhm.
something that's the holy grail. Any thoughts about visual acuity in these eyes?
Yeah, I think we in ophthalmology and retina in particular have become very spoiled by BCVA because we had such amazing results going back to the anti-VEGF era, now 20 years old, right? These drugs rapidly improved best corrected visual acuity on ETDRS measurements. And clearly it's a functional metric. And it's obviously, flawed. It has its own flaws. It's subjective. The variability is quite noisy within the same patient and from within the same patient from visit to visit. As you know, the variability can be 8, 9 letters from visit to visit in neovascular AMD and similarly in geographic atrophy. So it's a noisy metric, but nevertheless, it's a regulatory accepted metric. But I think increasingly people recognize that in GA, it's probably not a good metric. It's a very flawed metric for a whole host of reasons. It's noisy and subjective and highly variable. But beyond that, it's very, very susceptible to sort of random stochastic movements of the lesion. So you can imagine a very big lesion that spares the fovea and patients can have excellent vision forever. Or you can have a very small lesion that just encroaches onto the fovea one fine day and the patient's vision just drops a lot. So it's really not in any way correlated with the movement of the disease, the efficacy of the drug. It's more due to random chance. And on top of all that, people with very poor fixation, as GA patients tend to be, also have an added noise on top of this metric. And if you want to add one more problem with BCVA and GA, that's preferred localization, preferred retinal localization, right? The preferred locus of fixation. So patients can learn to fixate eccentrically, as you know, and so that can be wiped out or preserved very randomly within patients. So for all these reasons, BCVA is not a very robust metric. I think if you get lucky, you might see something. And so far, we've been, I guess, fortunate to see in the K8 trial preservation of visual acuity in these 20 patients up to 3 months. We actually see a slight increase by about a letter and a half. And in the control untreated eyes, we see a decline of about a couple of letters. So we're seeing the vectors moving the right direction, but we're not making a big deal out of it for the reasons I mentioned, because BCVA is a really poor and shabby metric in GA. We need, if at all we need, and maybe I'll take a little contrarian view, if we need a functional metric in GA, then BCVA is certainly not the best one.
Well, that's a great explanation, and the complement inhibition team will find it interesting as well because they've struggled with this issue.
Yeah, I mean, there are so many issues. And so, of course, people have talked about low luminance visual acuity, microperimetry, multifocal ERG. All these measurement tools have their inherent problems as well. So there's going to be a big challenge. I want to take back, if you permit me, there's a reason why the FDA accepted the size of the lesion growth as an approvable endpoint. And I think a lot of people may not be familiar with why that is. It actually harkens back to the Vitrasur trial with CMV retinitis. I know you remember that trial, but a lot of younger folks in retina may not be aware of that. Back when Wiley Chambers was head of the FDA during that period of time, he and the agency accepted that loss of retinal tissue is a bad thing, right? And that's how that CMV drug was approved. And is really a legacy of that trial that the FDA accepted GA lesion size in terms of RPE loss, loss of retinal tissue being bad, as an acceptable metric. So it's not like they invented something new here. It's they're continuing what they had more than 20 years ago. And I think it's perfectly reasonable to assume that loss of retina is a bad thing. Just because you can't measure it for all the sort of stochastic vagaries we mentioned with BCVA doesn't mean that a drug is not actually helping people.
That's a great explanation and a great segue into what the next steps that you're going to be taking in your K8 program.
Yeah, so I think we're very encouraged by what we're seeing at the 6 month endpoint so far. It's tracking very well like before, so we're very encouraged by the positive results we anticipate having by the summer of this year from our phase 2 trial of K8 in GA. And we're now making plans to start a pivotal phase 3 study in GA. And you'll notice I mentioned study instead of studies, given the new FDA default guidance as of 2 weeks ago, that their default position for approvable trials is going to be a single trial, a single well-controlled and adequately powered study.
How many patients do you think you'll have to enroll in the phase 3 trial?
Yeah, so this brings back the earlier point you mentioned. And so it is true that a bilateral study, which the FDA may or may not permit, could be done with far fewer patients than with an unrelated patient study. Say 100 patients or 150 patients versus 300 or 400. But unfortunately, that's not the actual rate limiting step. The FDA has long given guidance, the ophthalmology division, that they want to see 3 to 400 eyes for safety at or higher than the approvable dose of your drug. So even if you were able to show statistically significant results for efficacy with 50 patients or 100 patients, it wouldn't matter because you would have to show safety in 3 to 400 eyes at the dose you want approved or at a higher dose. So at the minimum, you will need to treat, we will need to treat at least that many patients. So I think, typically, while we haven't finalized the phase 3 study, I think we would look at a two-arm, low dose, high dose kind of approach, each with 3 to 400 patients and a positive comparator group. So that's what we're actually sort of coalescing around.
That's wonderful. Well, Jay, thanks so much for this discussion. We really covered lots of ground, and I think you've given us a good vision for what the future of GA therapy might be.
While we at Inflammasome Therapeutics are very excited about what we can bring to market and actually open up the possibilities for vision saving and vision improvement and disease progression stoppage using this novel approach that targets multiple factors for GA. Thank you.
Well, thank you, Jay.
Thank you.